The overall goal of this procedure is to illustrate a general protocol for slice, fast scan, cyclic voltammetry. This is accomplished by first preparing the brain tissue for coronal slicing. Then, electrodes are placed onto the discrete brain region of interest. The next step is to deliver voltage as a triangular waveform through the electrode to detect dopamine. Finally, a pharmacological agent of interest is applied to the brain slice, and its effect on presynaptic dopamine dynamics is observed. Ultimately, this protocol shows changes in presynaptic dopamine dynamics, such as stimulated dopamine release and dopamine uptake rates through fast-scan cyclic voltammetry.
Though this method can provide insight into presynaptic dopamine dynamics, it can also be applied to other systems, including animal and neurotransmitters, such as rats and fruit flies, and serotonin and norepinephrine respectively. Demonstrating the procedure will be Francis Maina, Madiha Khalid, and Apawu, the graduate students from my laboratory.
To begin this procedure, according to the accompanied manuscript, prepare 3 types of artificial cerebrospinal fluid solutions: sucrose ACSF, ACSF solution for voltammetric recordings, and modified ACSF solution for electrode calibration. Next, transfer 10 milliliters of sucrose ACSF into a small beaker and place it in an ice bucket. Then, place the instant Loctite adhesive in an ice bucket. After that, prepare a razor blade and the necessary tools.